Good morning, everyone, and thanks for coming to my talk. I'm Professor David Wood at Ohio State University, and for about the last 20 years, I've been working on a technology involving self-cleaving affinity tags. I'm in a position now where we've co-founded a company and are hoping to commercialize this technology in the next couple or 3 months, and I wanted to tell you a little bit about where it came from, what it can do, and where I think it's going. And so, what I wanted to do was just to get us all on the same page, do a very, very quick introduction into biopharmaceuticals, because this is really where we see this tag making a big difference. And so, as you know, biopharmaceuticals are very large drugs. They're polypeptides, much larger than conventional synthetic molecules. And as a result, they need to be expressed in some kind of a living host cell. Bacterial expression, E. coli, and mammalian expression of HEC and CHO are probably the two most common ones, but there's yeast and insect systems as well. So as a consequence of these complex molecules being made in complex cells, there's quite a lot of impurities that have to be removed before these drugs can be injected into a human patient. And really where our technology has its impact is in this purification, this downstream processing of the purification of the proteins. And of course, the main thing that we worry about is host cell proteins and getting rid of those, but really getting rid of all of these and getting the drug in a way that it can be injected into a human. And so a consequence of the biopharmaceutical industry, the large profit margins for these drugs, has led to a couple of important points that kind of dominate the thinking of the industry. And one is that the speed to market is absolutely critical. The way I explain it to my students is if you have a drug that's going to make $1 billion a year, that translates to roughly $3 million a day. So for every week that drug is delayed, it's costing basically the salaries of everybody who's ever worked on that drug. And so the speed to market is really critical. And part of what comes out of that is that the industry's become very conservative. So if there's any risk associated with the drug, they really don't want to take a lot of risks, and they like to keep their own platforms. There is some pressure to innovate, and as things change, that pressure is becoming greater. But right now, the industry would like to stick to the technologies that they know and that they know that they can get past the FDA. And those two things coming together create the third point, which is that a lot of these drugs are made using established platforms, especially for monoclonal antibodies. So if you have different drugs, they're all basically made the same ways. And some of the things that are changing now is that the cost of goods are starting to become important, especially as biosimilars are being released. And there's a lot of new modalities on the horizon with things like gene therapies and exosome delivery vehicles and new types of antibodies and antibody fragments. But if we go back and kind of look at the sort of big established platform, what we start with is a bioreactor that has our cells, whether they're E. coli or mammalian. This system that I'm showing you is a perfusion system, but one way or another, that cell broth gets clarified and put onto a series of chromatography columns. The first one is usually referred to as the primary capture. And so these chromatography columns, when I'm explaining this to investors and other people who don't know much about bioprocessing, they're like Brita water filters where you put a liquid through. The liquid has a whole bunch of stuff in it, like a vegetable soup. And if you're lucky, your Brita water filter will grab onto just what you want, which would be either your drug or everything that's not your drug. And that allows you to sort of allow a purification of the drug pretty easily. And so I know, given the audience, you already know all of this stuff, but I've gotten into the habit of kind of explaining where my thoughts are on downstream processing going forward. And so if we go back to the monoclonal antibody platform, this is the one that makes about 70% of the revenues in the biopharmaceutical industry come from monoclonal antibodies, even though if you look at the number of therapeutics, they only represent about a third of them. So they tend to be very lucrative. And what's the reason that there's a single platform for all these monoclonals, Humira, Enbrel, Rituxan, these things that you see on TV, is that they all have basically the same structure. So what gives them their identity is what we call the variable domains at the end. So those are going to vary depending on what drug you have. But on the other end of the monoclonal, there's what they call the constant domain. And the constant domains are all bound by protein A, which is this first affinity column that allows you to purify all of these different antibodies at 95% purity. So the point that I'm making is that these antibodies, because they have a single feature that is common to all of them, a single platform can be used to purify pretty much all of them. And then there's some polishing steps, but once you're at greater than 95% purity, the polishing steps are not too hard to create. And so if you look at protein A, because it's so effective, it's the current platform. And what a lot of people complain about is that the protein A resin costs quite a lot, $12,000 to $15,000 per liter. And at full scale, you have a column that's 2 meters in diameter and half a meter deep, holding 1,500 liters. And so the resin is about $20 million to do a single process. And so a lot of people talk about how expensive that is and how it's a huge problem, but there is a lot of thinking that goes into this. The resin can typically bind about 50 grams per liter, and it can be regenerated a whole bunch of times, upwards of 200 plus times. So over its lifetime, that 1 liter will purify about 10 kilograms of MAB. So the protein A resin, once you equal that all out and account for its reusability, it represents only about $1.50 per gram of MAB produced out of about a $300 to $100 total cost. And a typical MAB, remember, sells for $2,000 to $8,000 per gram. So the protein A resin, as expensive as it seems, is really a rounding error on a rounding error in terms of the cost of the protein. And as a result, this is a great example to follow of a really well-established platform that's used by pretty much every company that makes monoclonals. And when we first started our self-cleaving technology, people asked if it could be used with MABs, but what I realized over time is that only the worst kind of fool tries to go up against protein A. Although I will tell you, I've seen some very exciting talks about continuous precipitation. So the challenge that we had was not how to beat protein A. We're not even going to try to do that. What we decided is let's look at these 2/3 of all other therapeutic proteins that also have to be purified by some other platform. Now, the problem with these proteins is they don't have any common features. Those proteins that I'm showing you on the bottom half of the slide, they're all actual therapeutics. Tissue plasminogen activator is in there. You can see little insulin over there sort of on the left. And then there's Neupogen and Epogen and other drugs in that group. But they're proteins, but they all look different. So the question is, how do you purify them? And in a conventional process, what you do is you assume that you're going to have to do conventional chromatography. And you could assume by starting that you're going to do a 3-column process. And so let's say that for the first column that you're going to look at, for the first column in the process, you're going to look at an anion exchange column. Now, for that single column, you're going to have to optimize the column type, whether it's Q or DE, which vendor. You're going to have to look at the pH and the conductivity. You're going to have to look at residence time, different volumes, wash volumes, and you're going to have to look at regeneration protocols. And each one of these parameters is going to require a combinatorial set of experiments. There's very clever ways to do it with design of experiments. You can have robots do a whole bunch of experiments. But at the end of the day, you're going to end up doing this for every single column in this 3-column process. And each protein is going to require a certain column in a certain order where each column is optimized for that product. And at the end of all of this, it can take 6 months or longer to develop a commercial scale process if you don't have any kind of a common feature. So you have to start from scratch with each new protein. And this leads to some real problems with developing these drugs. It slows it down, it creates risk, which the industry hates. And so the question is, can we come up with a platform for everything else? A single affinity capture that would be able to purify all these proteins. And of course, those of you who work in the area of protein expression, especially at high throughput research and laboratory scale, you say, well, of course there is. We'll just put on an affinity tag. And so the affinity tag, you're cloning the DNA of the target protein into whatever cell you want to make that protein in. But then you add a little piece of DNA to it that creates a tag so that when that protein gets expressed, it has this little wrench ready to grab onto something. And you can go ahead and put tags on pretty much anything. So I'm just kind of flipping through, put a tag on all of these proteins, and now when you're done, all of these proteins now have a common feature. They all have a tag on it, and therefore they can all be purified using a single affinity resin that tag will grab onto. And so most of you have used the His tag. Some of you might have used the maltose binding tag or GST. Many tags available, many systems, the twin strep tag, I could go on and on. And this allows you, by tagging a protein, to purify really any protein you want using a single column, using a known protocol. So for high throughput, this is really necessary because you don't have 6 months for every new protein that comes down the pipe. But if you're in the biopharmaceutical industry, typically your target protein is a human protein like insulin or growth hormone or something like that, or a human antibody. And the idea is that if you use a human protein, you don't have to worry about an immune response. Like back in the day when people would use cow or pig insulin, and about a third of the patients would become allergic to it. The problem is the tag. The tag is not human, which means it is potentially immunogenic. So if you were going to try to use a tag-based method to make a protein that you wanted to later inject into a patient, you would have to remove that tag. And so the conventional way to remove the tag is to add a protease, and hopefully it will target a protease recognition sequence that you put between the tag and the protein. And then you have to do another process to get rid of the cleaved tag and get rid of the protease, although there's been a lot of progress on putting his tags on the protease to get it to be able to subtract it pretty easily. But at the end of that, you end up with your purified product. So you might say, well, the problem solved then. We're just going to put tags on everything and do that for clinical research. But here's the problem. The first is that when you go to scale this up, the protease enzymes are actually quite expensive. They have to be produced at GMP quality in order to be introduced to your GMP quality process. You still have to do the additional processing. But what's even more of a headache, especially in high throughput research, is that sometimes these proteases randomly chop up your product protein. And then every other time you add the protease and it just doesn't cleave off the tag. And you don't know why, and people talk about steric hindrance and other things. But it becomes something that, even though it works great and people do it at laboratory scale, proteolytic cleavage, especially with an endopeptidase like this, is never used in bioprocessing, ever. You will not find a drug where there's a protease removal of a tag. I heard a rumor once that there's one where they use an exopeptidase to get rid of a his tag, but I was never able to identify which protein that was. So even though some of you may know something that I don't, I think all of us would agree that this is an extraordinarily uncommon in manufacturing. So there's your problem. You need to use an affinity tag at research scale where you don't have 6 months to look at each new target. But at manufacturing scale, you can't have an affinity tag. So what often happens in companies is they say, okay, I'm just going to leave the tag on, and I'm going to do all of my preclinical studies with the tag. So I'm going to do my binding studies, my assay, my tox studies. But deep down, I know that if I go into clinical trials with that tag protein, I'm going to get blocked. The FDA is going to have a fit, and they're going to say, you got to get rid of that tag before you start having patients with immune responses. And the upshot of all of this is that if you are going from discovery to clinical manufacturing, in discovery, you're going to have tag-based systems. You don't really have a choice because you have too many proteins coming down the pipeline and not enough time to figure out a formal process for purifying them. On the other hand, when you go into clinical manufacturing, you're going to have to come up with a complicated new process that's going to take months to develop. And at some point between discovery and manufacturing, you're going to have to make that transition. And so this slide was made by my student, Joe Terrace, who's graduating soon and has done quite a lot of the work that I'm going to be showing you today. But I have a little picture of him because I blatantly stole this slide from him, and I figured I should keep his photo on it at least. But I'll talk about him at the very end. So that really comes into where my talk is. So the idea is, can we engineer a new universal purification platform, but that will create a tagless target and is therefore useful at research scales for high throughput applications where you need to look at a lot of different proteins in a hurry, but then can go seamlessly into manufacturing and deliver a protein that the FDA would consider safe. And so the story begins with the discovery of this kind of protein element called an intine. That's a self-splicing protein. So those of you who do a lot of molecular biology remember self-splicing introns. An intine splices at the protein level. And forgive me if you've already heard a lot about intines. When I was starting my PhD, intines had just been discovered. I started my PhD in 1993. And for the first 10 or 15 years of my career, I spent a lot of time explaining to audiences, even in biology conferences, what intines were. And somewhere along the way, it seems that they've gotten into textbooks, and now even the undergrads in my classes know what they are, and they sort of roll their eyes when I explain them again, but some habits are hard to break. But in any case, the intene can self-splice, and what's cool about the intene is you can move it to a different context and it will still function. It's completely self-contained, at least many intenes. And so pretty quickly, the splicing mechanism was figured out, and I'm not going to go into detail on that. But an upshot of that understanding of the splicing mechanism was the generation of intenes that, rather than splicing the two extenes together, could either cleave the extene from one terminus or cleave the extene from another terminus. And so the N-terminal cleaving is generally induced by thiol addition, and it's a chemical cleavage of a thioester that forms during the first step of the splicing, and then the splicing is arrested. So the thiol effectively cleaves a thioester bond. The C-terminal cleaving, on the other hand, is induced by a temperature or pH change. So it's spontaneous, but is sensitive at least a little bit to the environmental conditions. And so as a PhD student, I'd been working at Amgen for a couple of years making Neupogen, and I knew that adding 50 millimolar thiol to anything is not going to be scalable. So I decided to focus all of my efforts on C-terminal cleaving, and we came up with the big idea. And the big idea was that we'd still have our affinity tag, our binding domain, but now we'd add an intene between the affinity tag and the product protein. We could express that as a tripartite fusion protein, and then go ahead, bind, wash. But instead of eluting and then using a protease to cleave off the tag, what we would do is we would simply trigger the tag to self-cleave by the intene mechanism and release the product, now tagless, from the bottom of the column. And this was very cool. And in my PhD work, we started with our Mycobacterium tuberculosis intene. This is not a picture of it, but it's a picture of one that looks just like it. We deleted the homonuclease domain to create a mini intene to make the system more efficient. We went ahead and did some directed evolution using a genetic selection based on thymidylate synthase, and that genetic selection allowed us to find a cleavage mutation. And this figure is from my first author paper that ended up in Nature Biotechnology, which was pretty nice. And what the cleavage mutation does is it allows the pH sensitivity of the cleaving rate to become greatly accentuated. So what you see is the rate constant in the y-axis, the pH in the x-axis. And as you go from pH 8 to about 6, one of these intenes suddenly cleaves much, much faster. And that intene became known as delta ICM. And so delta ICM, it turns out, is also sensitive to temperature and pH. And so it gives you a lot of levers to pull if you're going to try to control the cleaving activity. And so the big idea was that you could express the protein at relatively low temperature in E. coli, where the intracellular pH is about 7.5. You get an uncleaved precursor protein, which then you could stabilize by lysing the cells at pH 8.5, do your binding and washing at pH 8.5, and then shift the pH, induce cleaving, and get your purified product out the bottom of the column in a single step, where really all you've had to do is change the pH, which is quite trivial compared to adding a protease. And so pretty quickly, we went ahead and published this intene, and here's a protein gel. In lanes 1 and 2, it's the whole cell lysate and the clarified lysate of a maltose with an intene, and the target protein in this case is human acidic fibroblast growth factor. We induced the cleaving reaction, in this case by putting a pH front slowly through the column and allowing the protein to cleave and collect as a peak. And then at the end of the process, we just add maltose buffer and we can regenerate the tag. Strip off the tag, regenerate the resin, and what you see is that the precursor protein is almost completely depleted, and all you see really left on the column is cleaved tag. But this gel shows sort of really high purity that you can get from a single intene resin, and we had underloaded the column, I will admit, because maltose tags tends to leach off a lot. But here's another example where we purified a subunit of RNA polymerase, and again, in this gel we have the time course. So in about 2.5 hours, we were able to collect a very highly purified product peak, again with maltose binding protein as the tag and our delta ICM intene. In this case, we raised the column temperature to 30 degrees to accelerate the cleaving reaction, but in 2.5 hours, we could collect a fairly nice, concentrated, and a highly purified peak from a single column. So we were very excited. We published a bunch of papers. And of course, we filed a patent, and the patent issued several years ago, I think 2005. And we were off to the races. We were all going to become millionaires. We found companies that were excited about this, and it was going to be great. Now, what we didn't know or we sort of knew, is that New England Biolabs was also developing their own intine-based technology that in this case was induced by thiol addition, and it became known as the impact system. And they were able to get this system to work, and they also published papers. And I have to admit that a lot of the early, really breakthrough work on the intine mechanism was done at New England Biolabs, along with a few academic labs. So they really added a lot to the field. But unfortunately, they also decided to file some patents. And if you've heard of the impact system and you've never heard of delta ICM, the reason is because they got to the patent office before us. And so if you've used any intine technology, you probably remember this figure from the brochure, and you've used the impact system. So I could give a whole other talk about how we were unable to come up with a reasonable license agreement with them. But in the end, they effectively blocked us from commercializing our technology, and in return, we didn't allow them to use our technology. So the impact system and eventually the P-twin system was sort of what you got, even though to this day I will still argue that our technology is probably better. So that's how things stood as I was graduating from high school, or not high school, from my graduate work. But there was still a problem that nobody wanted to talk about because we didn't know how to solve it. And the problem was that even though we had this great intine that had a really nice pH sensitivity, so the rate constant on the left here is actually log units. So you're talking about a huge difference in cleavage rate over pH and temperature. And in E. coli, it turns out the cleavage was pretty slow. It had a half-life of 17 hours. And of course, you express proteins in E. coli for a few hours. So if you have the right protein and you express it at the right temperature, typically you can get precursor. And you can purify it and you can get the cleavage reaction and everything's awesome. But I have to tell you, if I had a nickel for every time somebody said, is this going to work in CHO, I wouldn't have to write any grant proposals. And so the problem with CHO is that under the conditions of expression, where the growth media is pH 7 and the temperature is really close to 37, the intine wants to cleave much faster. And its half-life is about 1.5 hours instead of 17 hours. And of course, if you've ever done any kind of mammalian expression, you know that the expression takes several days, if not weeks. And so the result is that the tag has completely cleaved off of the target protein before you ever get it out of the cell culture reactor. So you'll do a great job of purifying the tag, but your protein's going to float away with all the contaminants because it's not going to be attached to the tag. So there we stood. Basic intines designs, impact Sion on the left, which we knew required thiol that would destroy disulfide bonds, was too expensive and too toxic to really scale up. On the other hand, we had the pH and temperature shift of our intine, but that led to a lot of premature cleavage. And of course, the thing that all of us didn't want to talk about and just swept under the rug was that it had both of them showed unpredictable cleavage behavior. So sometimes with delta ICM, your target protein would cleave completely, no matter what you tried to do. Other times it would cleave very slowly or not at all. And the same was true for impact CN. And so this is sort of where we left it. I graduated with my PhD. I went to work at Princeton. And for the next 20 years, I kind of played around with trying to make this a more attractive system. And so one of the things that we published was ELP tags. What ELP tags do is it's a long polypeptide tag that you can add just like an affinity tag. But if you add a little ammonium sulfate or raise the temperature, they fall out of solution and become insoluble in a very reversible way. So you take an ELP, you put an intine, you attach it to a target, and you can do a pretty decent purification. If you look at lane 6, we're purifying chloramphenicol acetyltransferase, basically in a single test tube by just precipitating, washing, resuspending, cleaving, and then precipitating the tag. And so this was pretty cool. We got that into Nature Methods. We showed that you can actually get pretty high purity if you wash the pellet a couple times and kind of optimize it. We showed that you can attach the ELP tag to one subunit of a larger complex. In this case, it's the E. coli RNA polymerase. And by just tagging one subunit, you can actually purify all of the other subunits. It looks like there might have been some disappearing, but the beta, beta prime, alpha, sigma, and omega all purified this way. And of course, we showed that you could use it for high throughput purification. And there was a lot of interest in this. A lot of labs requested it. And another thing that we went ahead and looked at is I wanted to try to figure out how to get this into the biopharmaceutical industry. And so I worked with a guy, Jesus Sanz, in Spain, who had developed something called the LITAG. And what the LITAG was is it's a choline binding domain from Streptococcus. But it turns out choline looks a lot like a quaternary amine. And so I thought, well, if it'll bind to choline, why won't it bind to a Q-sepharose column that I can get that's ready for a GMP process? And sure enough, if we put this LITAG, this choline binding domain, with an intine, we can get really nice purity using a DEAE column where the protein binds as if it were an affinity column. So extremely precise. You can raise the salt to something really, really high, knock everything else off, and then cleave the target and leave the tag, most of the tag, on the column. And again, we hugely overloaded this column to give you an idea of what kind of purity you could get. And the beauty is this was would be a DEA resin that you could buy from GE, or I guess now Cytiva, and go ahead and put straight into a GMP process. So ironically, we never published that. It's still sitting in an inbox. Everybody graduated. But we had all of these great ideas for how we were going to come up with ways to try to get this intine into the biopharmaceutical industry and hopefully turn it into a platform. And so 20 years later, this is where I am, and I'm going to tell you what we've been doing. Now, the problem is 20 years later, even though I'd played a lot with tags and I played a lot with sort of trying to figure out how to make the intine work in biopharmaceutical applications, at the end of the day, we never really solved these problems, where we either have a thiol intine that destroys disulfide bonds, or we have this premature cleaving that keeps you out of mammalian cells. And so what ended up happening was we got included on a DARPA project where the goal was to create a briefcase that could make any biopharmaceutical in 24 hours. So this would be something that you would carry around with you, and it was for soldiers on the battlefield. And if you needed a dose of insulin, you would just type in insulin, growth hormone, type in growth hormone, Humira, type in Humira, and out in 24 hours later would pop a single dose ready to inject of whatever drug you had asked for. And as you can imagine, if we would have to be able to purify these proteins, and so I was approached because people saw my intine technology and they said, hey, if you can really purify anything with these intines, then we want to use this technology in our DARPA briefcase to make all of these drugs. And it's a very exciting technology. The lead on the project was a guy named Govind Rao at the University of Maryland at Baltimore County, and he is still pushing that technology. I'm not going to talk about it very much today, but if you email Govind or Google Govind Rao or the DARPA Biomod project, BIOMOD, you'll get to learn all about it, and some papers were published, and it was pretty cool. But at the end of the day, these problems, the pH temperature shift, was still a headache for us, because even in the biomod system, we were still getting premature cleaving before we could do the purification. And so at that time, I had a visiting student from China named Changhua Shi. He goes by Stephen, and he's at Wuxi now. He's interested in returning to the United States, and I should tell you, if you can catch him, he's an incredible, incredible scientist. And he told me that, people were looking at these split intine systems, and there were naturally occurring split intines that were pretty soluble and behaved pretty well. But the beauty is they can't cleave until they assemble. So these are intines that are expressed in their native host in 2 segments from separate genes. They float around inside the cell until they find each other, and then they go ahead and they splice. And so the idea was that we could assemble these on the column, wash the column and cleave, and we could get rid of the premature cleaving. And so in order for this to work, we would need things like pH sensitivity, rapid cleaving, reliable performance. We need all the things that you want from a protein purification system. And we set about to re-engineer the NPU split intine. So we started with our basic idea where we just put a split intine where the existing intine was. The first thing that we did, and we were following the lead of a scientist at Texas A&M named Shaili Chen, we moved the affinity tag to the other end of the N intine. And this sort of tricked the intine into thinking that the first step of the splicing reaction had already taken place, and that releases the third step of the splicing reaction and greatly accelerates it. The second thing we did is we made the same mutation that we have in our delta ICM intine, which also accelerated the reaction. We neutralized a couple of cysteine residues, and I'll explain why in a minute. And we made a serine to histidine that would also help accelerate and make it pH sensitive. So basically, we were taking our entire knowledge base and applying it to this intine in a very rational way, because it turns out for split intines, directed evolution is hard. And what we found was this intine. So here's the precursor protein at hour 0, and at hour 16, you can see that it's cleaved. At pH 8.5, you get a little bit more than 50% cleaving at 16 hours, but at pH 6.2, you get a little bit less than that. So it's a little bit pH sensitive, but the truth is nobody's going to want to wait 16 hours at room temperature to have their intine work. So what we did was, and this is again a really long story, we started looking at the residues at the front end of the intine. And so we took that -1, the first residue before the intine, outside of the intine, and we changed it to all 20 of the naturally occurring amino acids. And then we measured the rate constants at pH 6.2 and 8.5. And what you realize is the identity of that amino acid has a huge influence on how fast it cleaves on the other end, but also has a huge influence on the pH sensitivity. And so we did some rational engineering and some mutant design, and we set up a FRET system that allowed us to track cleaving, and we did some high throughput screening. And we eventually settled on GD-GHG immediately preceding the intine. And we call this the sensitivity enhancing domain. And along the way, if you look at that previous slide, we came up with a bunch of different combinations of amino acids that give us really almost a continuum of different behaviors for the intine cleavage reaction. And we haven't even started mining that for what we might be able to do with those. But the sensitivity enhancing domain we put on the front end of the intine. And now, if you look at pH 8.5 in 16 hours, you see maybe 20 or 30% cleave. But remember, you're going to be doing your binding and washing at 8.5. You're probably going to have everything done within an hour or two, where you see very, very little cleaving. Meanwhile, at pH 6.2, you have about 90% cleaving in 5 hours. And so this is the target that we were looking at, of getting a really pH-controlled intine cleavage where we would eliminate all premature cleaving, but still have that good handle during the purification process. And so what we did is we took that intine, we added a his tag to the front of it, we added a now unique cysteine residue because we'd neutralized the other cysteine residues. And then we would use that thiol on the cysteine residue to covalently bind it to an affinity resin. And the idea is you would buy this affinity resin from us, and we call it the ICAP tag resin for intine capture tag. And we would sell you this just like you would buy a protein A resin or any other affinity resin. And then you, as the customer, would take the 35 amino acid C-terminal intine segment, and you would put it on the front end of whatever target protein you were trying to purify, and you would put it into whatever cell host you liked. And the idea was that you would then bind that to our resin. It would become pH sensitive, so you'd bind at pH 8.5, you'd shift to 6.2, and your protein would cleave ideally within 5 hours at room temperature, but I'm going to get to how you can tweak that a little bit as well. And so the first thing that we decided to purify, besides green fluorescent protein, which was a lot of our development work, was granulocyte colony stimulating factor. So this is filgrastim, which is the generic name of the drug Neupogen, which ironically is the drug that I used to work on when I worked at Amgen. So I know the protein really well, and I knew a lot about the conventional methods for expressing it and refolding it and purifying it. And so you see the binding phase where all of the proteins come through, the pH 8.5 buffer to wash out all of the contaminants, pH 6.2, and then we stop the column, incubate for 20 hours, and I'll get back to that, and then we elute the product, the cleaved product. And here's the result. So the purified GCSF that you see on the left, again, a single column process. This is a high sensitivity silver stained gel. It's a little bit overexposed even. But you can see the clarified lysate in the flow through, and you can see the purified GCSF in the elution bands in the middle. And then we had a non-reducing gel as well. But I would argue this is really kind of a nice purification for a single column. I would argue that it's more pure than even what you would get from a his tag. But this is actually bona fide GCSF, identical to the Amgen product. And so on the right side, you see the SEC that shows that it's monomer with very little aggregate. And then we did an activity assay that showed that the material was actually more active than the standard that we buy. But going back to the 20 hours, so any of you who work in processing are like, whoa, 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 we're not going to want to wait 20 hours, especially at room temperature. So what are you going to do about that? And that led to the other problem, which we had 20 years ago, variable, unpredictable cleaving rates. And so we tried a whole bunch of different proteins with this system, and what you see is that, some of them cleave faster or slower than others. So streptokinase cleaves pretty well, but green fluorescent protein in panel B in the same amount of time is maybe 60 or 50% cleaved. And so that led to really the key breakthrough in all of this. And this was a predictive model for protein cleaving. So the question was, where is that variability coming from, and is there a way for us to solve it? And that's where the next sort of key experiment came along. So in this experiment, what we did is we took GCSF, which we knew cleaved slow. It took 20 hours to cleave overnight. Now, it turns out the first 5 amino acids on GCSF are MTPLG. So what we did is we took those 5 amino acids and we put them on the front end of green fluorescent protein. And what you see in the graph is that the green fluorescent protein then behaves almost identically to GCSF in terms of cleaving rate. In a second experiment, we took streptokinase, which was known to cleave quite quickly and very well behaved. It starts with MIAGP. We put MIAGP on the front of green fluorescent protein. And sure enough, the MIAGP then behaves exactly as streptokinase. And so the implication was that the first 5 amino acids on the front end of the target protein have this profound influence on how the target protein behaves. And in fact, the target protein may not matter at all. And all these theories that had been coming along for 20 years about steric hindrance and the active site and all kinds of other things may actually not be relevant. So what we did is we took the native extine residues, which are CFN. That's what normally follows this intine in nature. And we put them on the front end of enhanced green fluorescent protein. And then what we did is we varied the first amino acid, the cysteine, to all of the other 20 native amino acids. And what we found was that sure enough, that first amino acid has a huge influence on the cleavage rate. So what you see in the y-axis is the cleaving half-life in hours, where aromatic residues, tyrosine, phenylalanine, cleave very, very slowly, whereas charged residues and small residues, alanine, glycine, asparagine, those tend to cleave much more slowly. And up to that point, slower and faster. Up to that point, we'd been using methionine on the front of pretty much all of our targets because most people, when they think of expressing a protein, especially in E. coli, think of it as being methionine. But it turns out that MT, which is the first 2 amino acids of GCSF, they cleave slowly, whereas MI, which are the first 3 amino acids of streptokinase, that cleaves much faster. And those cleavage behaviors, the slower for MT, the faster for MI, is consistent with what we saw in the +1 amino acids. So the suggestion was, it might be that the first 2 or 3 amino acids matter, but they might matter in a way that's consistent and sort of additive to how the amino acid behaves in the first position. So to test this hypothesis, what we did is we took our the CFN, we put it on the front end of the enhanced green fluorescent protein, but then we varied the first and second position relative to different families of amino acids. So if we did all 20 by 20, it would be 400 clones, and we didn't have the resources for that. So we did a 6 by 6 and did 36 clones. And we came up with these data. So each one of these panels has the same first amino acid, and each line within the panel has the same second amino acid. And I often refer to this slide as sort of the Rosetta Stone of why intine research has been such a pain for so long. What you see is that if you put an aromatic, a phenylalanine on the front end, pretty much everything behaves really well. On the other hand, if you put a proline on the front end, it abolishes cleaving and you're done. But with methionine, which was where most of our work had been done up to that point, what you see is the cleaving rates are very variable and have a profound impact of the second amino acid and probably a little bit of the third. And so this explains why we were getting all of this highly variable cleavage rate all of those years. And in fact, if you look at the sort of middle cleaving ones, A, E, and K, that are sort of the middle line, cleaving a little bit slowly, what you see is E, K, and A, E, and K all cleave on the slower end of the spectrum. Whereas if you look at the ones at the bottom, MMN and MFN, that cleave very, very quickly and are basically done at 5 hours, that M and that F are on the faster end of the spectrum. And so the idea was that we could predict how fast a protein was going to cleave based on its first 2 or 3 amino acids. And more importantly, if we knew a protein was going to cleave slowly, we'd be able to fix it. We had ways of doing it. And so with GCSF, again, it takes 20 hours to cleave overnight. The wild type starts with MTP, but now I can look at the data that I have and say, hey, you either want to change the first or second amino acid to phenylalanine. And sure enough, if you change the first aminoalanine from methionine to phenylalanine, you have FTP, and now you have a really well-behaved protein that cleaves quite quickly. And so this was actually the breakthrough that made me think that we were ready to commercialize this technology. And before you complain that if you modify the front of the intine, it's not, or the target protein, it's not native human protein, this is a table that we threw together pretty quickly where we have released approved drugs and their trade names, the actual N-terminus of the human native protein, and the N-terminus of the drug substance. And the underlined amino acids are the ones that have been modified in order to make the drug easier to manufacture or expressible in E. coli in the case of GCSF. And along with that, we have the proposed changes that would allow these drugs to change to work with our intine. And so the point is that the pharmaceutical industry has been changing the front end of proteins since the beginning. I mean, GCSF goes clear back to the beginning in order to make them easier to manufacture. And as a result, we can use this intine to make therapeutic proteins that are perfectly consistent with drugs that have already been approved, many, many drugs that have already been approved. And so if we go back to this new affinity capture resin, we have the pH sensitivity, check, with our sensitivity enhancing domain. We have rapid cleaving with the different mutations that we've rationally introduced. And what's really nice is we now have reliable performance. We understand why some intines cleave slower, why some intines cleave faster, and we know how to fix it, and we know how to predict it. And then finally, I won't go into the detail on all of these, but we're looking at high binding capacities that we're optimizing now. I think cheap is probably an optimistic word. It's not going to be cheap, but it will be in the same ballpark as a protein A resin or a fairly nice affinity resin. But it's also highly reusable. We've regenerated these resins about 50 times now without any loss of activity, and the regeneration protocol is just a mild acid wash. It's nothing exotic. And so we've gone ahead and patented these. We have several issued patents now. We have some others pending. And we went ahead and created this product. And so the glossy brochure, the ICAP tag resin, the customer makes their tag protein in their system, bind, wash, elute, and regenerate the resin. And so what I wanted to do to kind of wrap up was show you a few of the case studies that we've done. This was a slide that came from the very first run on the very first column that we ever poured. And what we're doing is we're purifying superfolder GFP out of E. coli. And what you see is the column itself in the lower right, and the little green fluorescent protein in the test tubes that came out, and a gel showing sort of the purity that we got. And again, first try with the first column that we ever poured, we got a nice yield, nice purity, and we were very excited about this result. So we started trying some other things. One is we tried to do streptokinase. What you see is that we have some protease problems. So we get a nice purity, but there's definitely some degradation bands down below it. And it turns out that we looked into this, and if you add protease inhibitors, your favorite cocktail of protease inhibitors, you can actually clean up the protease problems. But what's cool is because the intine actually has a different mechanism than any of the serine proteases that you're trying to inhibit, the intine will still cleave perfectly happily in the presence of protease inhibitors. So moving on, here's another example of GCSF that we did from a later optimization. Again, there's some bands that we need to get rid of. But my argument is if you put something that pure into a polishing step, you'd be able to clean it up pretty quickly. But then the exciting part comes. So in this case, we're purifying a single-chain antibody from XB293. So this is mammalian expression. We're able to get really nice secretion into the cell culture supernatant. And again, in the elution bands, I would argue that you would be hard-pressed to find a significant impurity. And the top is a silver stain gel, on the bottom is a Western blot. And so we were able to show that we can get good secretion. We're using the serum albumin leader sequence, but it seems to work quite well. We can get good expression of these proteins. And and then we went ahead and did mature interferon from XB293. So again, you don't need the leader sequence because the intine cleaves off. But the cool thing about this case study, even though we do have some degradation, we got good secretion, good expression, good cleaving, good purity. But what I like to point out is that in this case, we were able to actually make mature interferon with the true native human N-terminus. So it's even more human than the therapeutic protein based on interferon that you can buy. And so again, GCSF made in mammalian cells, another really nice purification. And then for the first time in this talk, we've actually managed to make EPO. So Epogen, epoetin, expressed again in XB293. And again, I would make the argument it expresses well. It goes into the supernatant. It's completely bound in the flow through. And then after the cleavage reaction, I would argue that that's a really nicely purified glycoprotein. And I will have to admit that when we strip the column, that's what the CIP is, clean in place, we see some aggregated EPO a lot, frankly. And we're still trying to figure out how to optimize the buffers to minimize that. One of the nice things about this intine is it seems only to care about pH. So it's impervious to pretty much any other things that you would add that won't completely denature it altogether. So again, a really nice purification of a glycoprotein made in mammalian cells. And when we ran that through a mass spec, we found that the peak is a nice sharp peak on mass spec. And it appears that the peak is within the range that you would expect for glycosylated EPO. And so to wrap up, this is the non-protein A platform looking forward. We believe that we can now put an intine tag on just about anything. A lot of you are going to say, well, I have a protein that won't express, won't fold, won't stay stable, and is a total nightmare, and I've tried every tag in existence. This tag may not help you. It's really just to simplify the purification. The rule of thumb is that if you can purify with a His tag, you can probably purify with an intine tag. That's been our experience. If it's something that absolutely won't behave, then the experience may be different. But these therapeutic proteins, most of them are made solubly in either mammalian cells or they're refolded in E. coli. We're working on protocols to refold intine tag proteins, but we haven't run into any successes or failures yet. And we're not that good at refolding proteins, to be perfectly honest. So if you're good at refolding proteins, we would love to work with you on that. But the idea is now you can, in the research, you can put a tag on. You can purify things quickly, conveniently, high throughput. But that same first purification that you use to purify your first candidate the first time can now be scaled completely up into clinical manufacturing. The same column, the same process can be used to make a drug that you can now inject into humans because it'll be fully native with maybe 1 or 2 amino acid changes at the front end, but it will not have anything resembling a real tag on it. And so agile, low capital investment, and readily platformable. And just to wrap up, the company that we've started, and I need to put back in my disclaimer, since I have Ohio State all over this, I have to disclaim and let you know that I'm also involved with the company. And so Ohio State will not back me up on anything that I've said here. But the company that we've started is called proteincapturescience.com. I know that it's a mouthful, but try finding an unused URL these days. And this one was actually still free. And we are hoping to launch this product in the next 2 to 3 months, May, June, probably at the latest. And so we're looking for people who want to try it out. And we've budgeted for some free samples. Don't ask us for a liter of resin. But if you want a 5 ml cartridge, like a high trap cartridge, that can be arranged. And we're going to have 1 ml cartridges that we can definitely arrange. And if your protein binds and cleaves and you become interested in this technology, then we're hoping that you'll become one of our first customers. And so if you go to Protein Capture Science, we're still working on the web page. It's a little sketchy, I agree. But by the time we launch, it's going to be fully developed. But there is a sign up. If you want to go ahead and type in your email address and write any questions that you have, they come straight to me. And generally speaking, I'm very responsive. And so I just wanted to wrap up really quick by acknowledging my group. The big names on here, everybody has helped in some way in different pieces of what I showed you today. But the big one under collaborators and co-inventors is Changhua Shi. He was one of the ones who really convinced me to look at split inteins in the first place, and he did a lot of the early development work. And in my lab right now, I have Joe Terrace, Jacqueline Miozzi, and Brian Marshall, who are developing the product itself and helping me get it out the door. Before them, Michael Kulba, he's now at Sanofi. Sue Chang is at Regeneron. Sam Stimple is at, I believe, Sanofi. Also, Ashwin is at Merck, and Yaman is at Biogen. And so my students have done really, really well finding jobs. And honestly, if you're interested in hiring a top-notch PhD or undergrad, you should also. My students are doing extremely well. And I'm very proud of all of them. So that wraps up my talk. And what I wanted to do is thank you again for coming. I hope that you're as excited about this technology as I am. I do think it has the potential to make a significant impact. And I understand that if you post questions, they will be emailed to me. And I can tell you that I will be very responsive in answering them. So please email me if you want to set up a conversation. I'm happy to talk by phone, or we can even set up a Zoom meeting. As a professor, I'm really, really good at that now. So that's all I'm going to say today. And thanks again for coming. And I hope that I will hear from you soon.